Welcome dear viewers to the video on extended release drug delivery system for oral route. This is part 1 of the video, and many more video I will be going to make on this topic. This topic is little bit difficult to understand by many of the professionals and students, and that's why I have made the video. I request you to be with this video till end so that you will get the great understanding and in-depth knowledge about the extended release drug delivery system. In this video, we are going to see introduction to the ER formulations, or you can say it as modified release formulations. What are the different terminologies used, like extended release, sustained release, controlled release, prolonged release, and other type of modified release formulations. Then we will see advantages and disadvantages of extended release formulations. Drug properties required for making the ER formulation. That means what are the ideal requirements for the drug candidate to be formulated into extended release type of formulation. And then we will see what are the examples. So let's start with the video with the introduction part. See, as we know, oral route is the preferred route for drug administration because the patient can take those medicines by himself or herself. There is no requirement of technical person for the dose administration. So self administration is possible for the oral route by the patient, and it is easy to administer. Then oral route may be used for introduction or administration of the formulations like immediate release, sustained release, solid oral formulations, capsules, soft gelatin capsules, liquids like oral solution, suspension, and many more. But out of these, some disadvantages are there with the IR formulations. There may be the tablets, capsules, powders, or solutions or suspensions. Immediate release formulation may be required to be taken frequently because of the shorter half-life of the drugs and to maintain the plasma concentration of the drug in the therapeutic range. This is one of the reasons for making the extended release type of formulations. So extended release formulations are made to reduce the dosing frequency so that once a day or twice a day administration will be there, and it will reduce the pill burden. Extended release formulation release the drug at a slower rate or controlled rate over a longer period of time, and these characteristics of the extended release formulations make them unique and advantageous over the IR type of formulations. The extended release formulations may be tablets, capsules, pellets, or multi-unit particulate tablets. Sometimes the oral suspensions are also being tried to be in the ER form. So different formulations are there which can give the extended release profile with oral route. There are some terminologies used like slow release, extended release, modified release, sustained release or sustained action type of formulation. Then prolonged release, modified release, controlled release, or timed release. So generally all these terminologies are coming under the simple terminology for modified release formulation. Modified release formulations are those formulations in which the release of the drug is altered. It is not like IR formulation. it is modified, it is altered, it may be delayed, or it is tailored to meet the physiological requirement. I have made a video to differentiate between these terminologies. It is given onto the screen. The video name is Differences between Sustained, Modified, Controlled, Extended, Delayed, Prolonged Release Formulations. So you can visit that video to have good understanding about the terminologies. But for this video, consider these formulations as modified release formulations. So first is controlled release. In this controlled release, the drug release is in a slow and controlled way over a longer period of time, and the aim is to maintain the drug plasma concentration level in a therapeutic window. The drug release happens from the formulation and it is absorbed from the gastrointestinal tract in a slower way or slowly. Seer formulations are made with drugs having the short half-life. So shorter half-life drugs, if these are formulated into IR formulations, then to maintain the therapeutic plasma concentration, the IR formulations are required to be administered frequently. So controlled release formulations are there for drugs which are having the shorter half-life. Then come to the slow release, sustained release, or sustained action or prolonged release. So in this type of formulations, the release is slow over a longer period of time. And these are designed to avoid the side effects of immediate release. The side effects due to immediate release or burst release of the API may be nausea or vomiting or diarrhea or GI intolerance. So these formulations are designed to improve the patient compliance by single dosing or by reducing the side effects. Then coming to the extended release formulations. These provide even slower release over an extended period of time. These are slower than the sustained release and controlled release. You can understand and remember like this. These are having the more extended time period. These formulations have loading dose and maintenance dose. So loading dose is there to achieve the required plasma concentration to start the action, and maintenance dose is to maintain the drug concentration in the therapeutic range over a required time. For example, you are having an extended release formulation of, let's say for example, 100 mg, and in that loading dose is 25 mg and consider maintenance dose as 75 mg. So the loading dose and maintenance dose is calculated from the IR formulation, the physicochemical properties of the API, and the plasma concentration required to control the disease condition. Sometimes the ER formulations are designed with IR plus ER release profile also. Sometimes bilayer formulations are made, or the different population of pellets is used in the capsule, and by this the extended release profile is made. ER formulations reduce the dosing frequency and side effects by reducing the plasma drug level fluctuations. These formulations also improve the bioavailability of the drug and reduces the dose in the some cases. Extended release formulations are also advantageous for the manufacturers and researchers, as these can give you the intellectual property in the form of patents and copyrights or trade secrets. Also, these can give the new life to the existing APIs. So sometimes product life extension or hybrid type of formulations are made with the ER design or modified release design. So now coming to the advantages. So these give longer therapeutic effect or action, improved patient compliance by reducing the dosing frequency and pill burden, and the treatment will be better. Avoid side effects of IR formulations like high plasma level in a shorter time and reduce the toxicity. Modified release formulations or extended release formulations reduce the GI side effects and improves the GI tolerance. Some diseases are chronic in nature and daily dosing is required for treating or controlling those diseases like diabetes, epilepsy. So, if there are GI intolerance, then the patient will face GI problems like nausea, vomiting, diarrhea, and that's why patient compliance will get reduced. So, for such type of APIs and disease conditions, extended release formulations are better. These help in reducing the toxicity on chronic treatment and drug use. Then some limitations are also there, some disadvantages are also there, like dose dumping, loss or change in the ER release profile, which may lead to toxicity. Manufacturing cost and complexity is higher compared to the IR formulations. The toxicity may get increased sometimes and it is difficult to terminate the action. A release may get affected by GI conditions like fasting fed food conditions and presence of alcohol. So alcohol dose dumping study is required. Difficulty in swallowing of high dose tablets, capsules by patients if the formulation is bigger in size. Many of the drugs cannot be formulated in ER formulations. There are some formulations which came into ER formulation in the market and these formulations were withdrawn from the market or these formulations has not proven any advantages. So sometimes some drugs are not good candidates for the ER formulations. Sometimes some ER formulations failed, that's why withdrawn from the market. So these things can also happen. Then disadvantage is less flexibility in the dosing and administration. See, these ER formulations are not to be splitted or broken tablets or break line with the tablet formulation. So, if there is limitation of giving the break line or flexibility in the dosing, then these may have some limitations. So, dosing and administration with the split part is very difficult. If the tablet formulation is in the design of MUPS formulation, then the break line and dosing can be done. So these are some of the limitations. Then coming to the drug properties, how to select the drug or how the drugs are selected from making the ER formulation. What are the ideal conditions or what are the ideal drug properties or what are the ideal drug candidates for designing the ER formulation. So first is dose. What is the dose of the API required for the disease treatment? What is the single maximum daily dose? And what is the single time dose? What is the dosing frequency? So these are very important parameters to be considered. High dose drugs are there which are given in 1 gram or 1.5 gram like metformin. So there is limitation of the dose. If dose is high, that time it will be difficult to formulate that high dose API into extended release type of formulation because of the limitation of the tablet size or capsule size, swallowing difficulties. High dose drug with high solubility and high dose drug with low solubility. So these both situation will be difficult for the ER formulation. If the dose is high and solubility is also high, then there will be chance that higher concentration of polymer or release controlling polymer will be required. If the dose is high and solubility is low, that time it will be difficult to control the release through the polymeric system, and drug itself will have low solubility, and that's why it will lead to slower release. If the insoluble API is formulated into extended release type of formulation, then it will be very difficult to have the release profile similar from batch to batch and from lot to lot. So this is the limitation and requirement of dose. So there is no such ideal dose, but dose should be less than 1000 mg at least, so that the extended release formulation can be easy to formulate. Solubility and BCS class is the second point. Soluble and insoluble APIs can be formulated into ER design. It is not like that you cannot formulate the insoluble API or you cannot formulate the soluble API. It depends on the physicochemical properties, pharmacological properties, and the plasma concentration required. Based on that, the release profile required. Highly soluble drug may be difficult in controlling the release rate. Very low soluble APIs are not suitable for controlling the release. Very low soluble APIs itself dissolve at a slower rate, so making the ER formulation may not be a good choice. As you make the ER formulation of low soluble API, it will lead to less bioavailability. BCS class 4 needs to be formulated by increasing the solubility or solubility enhancement techniques. So generally IR formulations are preferred. Then stability in the GI fluid. So as a general requirement, degradation in the GI may lead to toxicity or impurity generation or loss of the drug effect. So drug should be stable in the GI fluids. Low bioavailability of the drug if it is getting degraded in the GIT fluids. GI stability is required for the drug to be formulated into ER design. Hydrolysis, degradation and metabolism at the site of absorption also makes the drug unsuitable for ER formulation. Protein binding and tissue binding. Now, drugs which have higher tissue binding or protein binding, which are higher lipophilic, these are poor candidates because these will have the higher or longer half-life. And that's why if you make ER formulation of these high tissue binding or highly lipophilic APIs, it will be of no use. Therapeutic category and therapeutic window. So generally the drug to be formulated into ER formulation should be having a wide therapeutic window. These should not be narrow therapeutic index drugs, so that you can avoid the toxicity or loss in the effect because of the ER profile and ER profile variations. So drug should be selected from the non-NTI category. Coming to the half-life, it is very important to consider the half-life. Generally, shorter half-life drugs are suitable for making the ER formulations, but there are some examples in which the half-life is medium or higher and still ER formulation is made. So half-life is one of the criteria, as ER design can reduce the dosing frequency of drugs with short half-life and compared to the formulated the same API in IR design. Half-life up to 5 hours is the limit or general limit. You can select the drugs which have half-life of 2 to 4 hours. These are the desirable candidates for making the ER formulation. Log P and lipophilic nature. See, I have made a different video on log P and absorption. So you can go to that video and you can learn how the log P and lipophilic nature affects the drug absorption. but for this video you consider that partition coefficient should be optimum, like near by 2 to 3 or 1 to 2, so that the drug can be absorbed by the oral route. Low log P drugs are undesirable as these are high hydrophilic. High log P drugs are also undesirable as these are highly lipophilic. Absorption site and mechanism of absorption. Drugs with specific site of absorption in the GIT are poor candidates for ER formulations because the ER formulation release the drug over a longer period of time, and if the drug is released and not absorbed, then that drug will be stagnant in the GI. Drugs with carrier mediated transport are poor candidates because saturation may be there. Drugs with carrier mediated transport are also poor candidates because saturation may be there. Drugs which are absorbed by passive diffusion, over transport, and throughout the GI tract are suitable candidates. So, if the drug is having good permeability throughout the GI, then it will be a good candidate. Also, the drug should not have pH dependent solubility. Otherwise, if the drug is released throughout the GI, it will be absorbed from the pH region where it is soluble. It will make difficult to understand the mechanism of release and the absorption. Then pharmacokinetic nature of the API. It involves absorption, distribution, metabolism and elimination. So till now we have understood the physicochemical properties. Now we will see the pharmacokinetic properties. So first is absorption. Rate of absorption of the drug from the GIT is key parameter for designing the ER formulation. Rate of drug release and rate of drug absorption are very crucial. Release rate should be slower than the absorption rate so that you can avoid the accumulation of drug in the GIT. For example, if you are releasing 1 mg per minute or 1 mg or 1 mg drug per minute, and if it is not getting absorbed, then it will stay in the GI and it will behave like a IR formulation because dose is done but still it is not getting absorbed. So always there should be a release rate slower than the absorption rate. If absorption rate is too slow, then ER formulation may release and transit the GIT. Thus, ER advantage is not achieved. Mostly the ER formulations have dissolution release profile of 8 hours, 10 hours, 12 hours or 16 hours, sometimes 24 hours based on to the dosing and the dose requirement. Low absorption may be attributed to the drug properties like solubility, pH and others. The release profile is to be set based on to the absorption rate and the plasma level required for controlling the disease. Distribution. Aqueous solubility of the API or drug, log P, protein binding, tissue binding, volume of distribution and elimination rate are the key parameters affecting the level of drug in the plasma, and that's why affecting the action of the drug. So drug distribution and elimination are required to be considered for ER design. Drugs with a high volume of distribution are poor candidates because if there is a high volume of distribution, that time drug will have longer half-life. First pass effect and metabolism. So first pass effect and metabolism variations will lead to variation in the bioavailability. Drugs which are undergoing the first pass metabolism and with higher metabolism rate are poor candidates. Drugs prone to pre-systemic metabolism, that means metabolism before absorption in the GIT, are the poor candidates. So drugs should be not much sensitive for the first pass metabolism and metabolism in the GIT. Then elimination. So drugs which are having higher elimination rate will have shorter half-life. So elimination rate is to be considered. The drugs with short half-life are good candidates for ER design. Drugs with longer elimination rate or longer elimination time and slow elimination may be problematic. The examples uh, for the ER formulations or prolonged release formulations are many. Some I have listed here like metformin SCL ER tablets, verapamil ER tablets, naproxen ER tablets, pelodipine CR tablets, topiramate ER capsules, pregabalin sustained release tablets, carbamazepine prolonged release tablets. See here, sometime in the European region, sustained release is called as prolonged release. So SR terminology may be similar to the prolonged release terminology in the European region. So this is regarding the ER formulations, and I hope you have got good understanding out of this video. I am going to make other videos to cover this topic completely. See, ER and control release type of formulations topic is a very big topic. It is a vast topic, and if you want to learn something about these formulations, you should have patience. You should have time to watch these videos, and slowly by repeatedly watching and reading the literature, you will get the good understanding. See, there are very less professionals or number of professional or people who know the ER formulation in detail, because the ER formulations are itself less in number compared to IR formulations. So I hope you will uh, uh, do like, share and subscribe to this channel, and also I hope you to comment onto the question onto the topic, and also post the questions in the comments. So let's meet in the second part of this topic, and let's discuss some good terminologies and good uh, information in those upcoming videos. Thank you for watching the video.